for your time. And just a couple notes related to the agenda. Evan is not presenting today because he had a conflict with coaching his daughter's softball game. So we will table Evan's portion of the clean water service provider and the functioning floodplain to a later date. And otherwise, I will give opportunity for public comment. I do recognize that there's some guests on today joining us, and if there's anyone who has anything they want to share up front, feel free to. Actually, sorry, before we do that, let's do introductions real quick. I'm jumping ahead of myself. So I'll just go in order of the screen, but I'm Karina Daly. I'm the chair of the Vermont CAC, and I live in Jericho, and I'm representing the environmental scientist position on the CAC, and I work for Vermont Natural Resources Council. And I'm looking for my vice chair, Denise, and it doesn't look like she's on yet. So I will go to everyone else as I see them on the screen. Breck.
Good evening, folks. Breck Bowden, member of the Citizens Advisory Committee from Burlington, former faculty member in the Rubenstein School.
Andrew.
Yes, good evening. Andrew Milliken, a member of the CAC from Charlotte and fairly recently retired from the U.S. Fish and Wildlife Service.
Hillary.
Hi, Hillary Solomon, District Manager for the Pulteney Meadowy Natural Resources Conservation District, and I think I represent South Lake of Lake Champlain.
Allison.
Hi, everybody. Oh, hang on. There we go. My headphones are doing weird things. Do you hear me now?
Yeah.
Okay, sorry about that. My name's Allison Spazek. I am also a member of, resident of Jericho, like Karina, and I think I'm a member at large on this committee. So, yeah,
and
nice
I
to see
think
everybody.
you are a youth member to begin.
Oh, yes, yes. I represent the youth faction.
Which is cool. Eric.
Eric Clifford, member of CAC from Starksboro and dairy farmer.
Lori.
Lori Fisher, non-youth member. I'm a resident of Williston, Vermont, and fairly recently retired from the Lake Champlain Committee. Hello, everyone.
Awesome. And Bob.
Hi, I'm Bob Fisher. I live in Barrie, and I'm the Water Quality Superintendent for the City of South Burlington. Thank you.
Mary.
Mary, and I work at the DEC in the Clean Water Initiative program.
And then guests, it looks like we have Karen Bates.
Karen Bates, the Agency of Natural Resources and the Water Investment Division, a basin planner for the Missisquoi, Lamoille, and Upper Lake Champlain basins.
Thanks for joining us. Ethan.
Yeah, good evening, all. I'm the Watershed Planning Program Manager with the Vermont DEC in the Water Investment Division, and really just wanted to be here to listen in and just continue to support the Clean Water Network. And congratulations to Lori on your retirement. That's really cool.
And Paul.
Thanks, Ethan. Good to see you as always.
Hey, good afternoon, everybody. This is Paul Trahan. I'm a newly board member for the Friends of Northern Lake Champlain, and happy to be here this afternoon.
Exciting. Thanks for joining us. I think that's everyone other than our guest speaker, Ellen. So Ellen, go ahead and introduce yourself.
Hi, folks. I have a slide in my talk saying a little bit more about myself, but I'm at UVM. I'm a researcher. I run the Wildlife Pathogens Lab, and I'm an angler, and I'm really excited to share our research today on heterosporous parasites in Lake Champlain yellow perch.
Thank you so much for joining us. Okay, so other than that, are there any other public comments before we move on? I did want to open the floor for that. Okay, and so I see Sarah Coleman has joined us. Sarah, do you just want to introduce yourself briefly?
Can you hear me?
Yeah.
Great. Good evening, Sarah Coleman from the Clean Water Initiative Program in Vermont, DEC, calling in from Syntaxis.
Thanks, Sarah. It's a little muffled, but we could hear you.
Okay.
And as far as updates on my level, I don't think there's anything that's not in the agenda that I can think of at the moment. So I think we can pass over that, other than the change in the agenda that I mentioned with Evan. And then our action item is to approve the meeting minutes for April, the meeting summary, or make changes to them. So, Lori.
I'm just looking at attendees here, Karina, for what I texted you about earlier.
Yes. And
do you want me to raise is it safe to?
Yeah, I think it's safe to raise the issue unless you think we need to go into executive session to do so.
Let's do that just in case anyone joins the meeting.
Do I need to have a motion to go into executive session?
Or we could deal with the minutes at the end of the meeting, as long as they're not distributed more broadly. Can you just clarify, or Mary clarify, that heretofore they've only been sent to the Vermont CAC members in draft form rather than a Vermont CAC email list?
That's correct.
Okay, great. Yeah, so why don't we do this at the end of the meeting? That way we won't have to have anybody who's a guest presenter come in.
That makes sense.
Okay.
Thank you, Lauren.
Thanks, everybody.
So with that, I will pass it over to Ellen for her presentation.
Great. Okay. I'm wondering how I should share my PowerPoint talk. I can send a request to share, and then I'm going to share this. Okay. Can everybody see my cover slide? Awesome.
Yeah.
Great. Okay. I guess I'll start presenting and then we'll open it up at the end for questions.
That's perfect.
Great. So I'm excited to share this work. Let me just see if I can get oops. Can you still see my slide? Okay. On the diversity, distribution, and prevalence of emerging heterospores parasites in Lake Champlain yellow perch. And this is definitely a group effort. I'll be talking about my collaborators. So just a little bit about myself. I'm at the University of Vermont. I run the Wildlife Pathogens Lab. We have a number of different research projects on a number of different wildlife hosts. A lot of them are vector-borne diseases, including tick-borne diseases. So you can hit me up another time for a talk on tick-borne pathogens, or we also do a number of other cool parasite and pathogen projects. I'm also run the internship program at the University of Vermont Natural History Collection. You may not know, we have a natural history museum on campus. We do, and we're actually trying to grow infrastructure so that we can start archiving fish in our collection, which we currently do not in the state of Vermont have any program where fish samples or fish themselves are archived. That's something that we're starting to launch. And then I also teach a number of different classes on campus. So this project is highly collaborative, as any well, most projects are. So it was really spearheaded by Tom Jones, Vermont Fish and Wildlife, as well as Matt Bodnar, Vermont Fish and Wildlife, who's now actually a pre-vet student at Cornell University, which is amazing. This work has been largely done in part by an undergraduate student here at UVM, Emma Privet, and also with Sarah Helms-Cahen, who is here in the biology department as well at UVM. And then we have a collaborator down at UNH at the New Hampshire Veterinary Diagnostic Lab, Inga Seidor, who does the histopathology, which we'll talk about later on. You're probably all really familiar with the growing number of threats to Lake Champlain fish, and you've probably heard about many of these different issues like contaminants. We have mercury and PFAS, pollution, invasive species. You've heard about aloes and round goby threat, toxic cyanobacteria blooms, and what I'm really passionate about and focused on are pathogens. And so these can be anything: parasites, fungi, viruses, bacteria, you name it, they all infect fish in Lake Champlain. And if you look worldwide, emerging infectious diseases in fish are on the rise. And we all know this is happening in mammals, including humans, but it's also happening in fish, which we hear less about. And these are diseases caused by a number of different pathogens, and they pose threat to aquaculture as well as fisheries. And so disease is not happening in by itself. We know that you have both host and pathogen involved in any disease system, but the environment is also involved. And so this is sort of like a One Health perspective in looking at the emergence of disease. It's really a number of variables playing in together. So for the host, it could be immune dysfunction, it could be stress, it could be any alteration in the distribution of a host. In terms of a pathogen, it could be a novel pathogen, or maybe it's just a pathogen that's already endemic that's increasing in incidence. And then in the environment, there are a number of things that increase disease incidence. So higher temperature of water. We have, contaminants lead to increase in disease, deoxygenation, sedimentation, etc. So all these things need to be taken in consideration when thinking about emerging infectious diseases in any host system. And then you're probably all familiar with all the crazy things that are kind of coming out and starting to be shown in Lake Champlain fish. These are some pictures by myself and then from a student, but you're probably familiar or have heard with largemouth bass virus, which first showed up in Lake Champlain in 2002, or lymphosarcoma in northern pike in 2002. Blue spot disease, we don't know the introduction date for that one, but what we study is heterosporous parasites, and those showed up in yellow perch in Lake Champlain, and only Lake Champlain, fingers crossed, in Vermont in 2014. And another common pathogen is columnaris. That's pretty common, and that's not only restricted to Lake Champlain. So what are heterosporous parasites? They are microsporidians, which are these really cool spore-forming, almost like fungal-like unicellular parasites that infect a wide range of actually vertebrate and invertebrate hosts. In fish, they've been reported in aquarium species, and fish are exposed to the parasite by consumption of spores, either through consuming an infected fish or through ingesting spores that are in the water column. So heterosporous parasites first were encountered in North America in wild yellow perch in 2000 in Wisconsin, in Wisconsin and Minnesota lakes. And at that time, the source of infection in the Great Lakes is unknown, and it's still unknown where these heterosporous parasites may have come from. And so the species described from the Great Lakes fish was described as Heterosporus sutherlandii. And what you see is an infected yellow perch where the skeletal tissue has been filleted, and you can see a beautiful cross-section of the skeletal tissue with infected and non-infected tissue. The infected tissue has a freezer burn appearance because the spores actually rupture the skeletal muscle and cause it it causes it to liquefy. And so you have this freezer burn or white appearance. So if anybody in here is an angler and you've been in the northern part of the lake and you've cut open and filleted yellow perch, which you do when you eat them, oftentimes you see this parasite. For me as a parasitologist, this this project with this system is really fascinating because for so many hosts, we don't get to cut them open and see the infection. So in yellow perch, you do a quick fillet, and right away you can see that the parasite is taken over. And so this is really damaging to skeletal tissue. And most anglers just throw their fish away. If you're at a seafood market, you discard the fish, you can't, it's unfit for sale. And extreme cases lead to kind of a concave appearance of the fish. And it's believed that it leads to indirect mortality because the fish aren't able to swim as fast. They may not be able to catch prey, and they may be more likely to be predated by other predators in the lake system. So Heathersporus sutherlandi was first detected in the Great Lakes in 2000, and since then it's been documented in over 32 water bodies in the Great Lakes region of the US and Canada. And in the lake study, it's been found in between 1 to 30% of fish sampled are found to be infected, with the majority of sites and studies indicating a 20% infection prevalence. And so what fish species are infected with Heterosporus in the Great Lakes? So far, it's not just yellow perch, but walleye, northern pike, trout, burbot, pumpkin seed, rock bass, and sculpin. And then laboratory exposures have found high rate of infectivity in rainbow trout, coho, salmon, brook trout, brown trout, lake trout, white suckers, mosquito fish, channel catfish, fat-hood minnow, and largemouth bass. So experimentally, a lot of different species can become infected, and naturally a lot of species have been found infected. So what is the history of Heterosporus parasites in Lake Champlain? So it's just a matter of time before we get here because we're connected to the Great Lakes. So it was first documented in Grand Isle in 2014. And then it was first found in Milton in 2017, and then in Georgia in 2018. And since then, it's just been found in increasing number of bays and fish. And so this is evident from anglers themselves, as well as fish brought in to be sold at Ray's Seafood Market. And so what do we know about Heterosporus parasites in Lake Champlain? We know that they're well established in our yellow perch populations, and they have been also visually detected in Lake Champlain walleye. And it's gotten to the point where we on the Vermont Fish and Wildlife page, they have a number of different pathogens to keep an eye on for anglers. And there's a Vermont fish health fact sheet on Heterosporus, just letting people know this is what it is if you see these opaque skeletal masses in yellow perch. And in terms of fish in Lake Champlain, you're probably all familiar with yellow perch and their importance in the Lake Champlain ecosystem. So they are key species for both recreational and commercial anglers. And so surveys have shown that ice fishermen and women typically target yellow perch in the winter. Yellow perch is the only fish harvested commercially from Lake Champlain. And if you think about the mass of fish taken out of Lake Champlain and consumed by humans, yellow perch is the #1 species. So approximately 400,000 pounds are caught by local anglers and sold. And the number of pounds that's harvested and consumed by humans is even more. So we eat a lot of yellow perch. So this is such an important species. And then unfortunately, because of invasion of white perch and alewives and other species, they've received significant encroachment into their ecological niche. So it's really important that we consider that as well for this species, yellow perch. Plus it's one of my favorite species. Just they're just fun. They're easy to catch. Yeah. Research objectives. So we set out to, with collaborators, document the distribution and prevalence of Heterosporus parasites in Lake Champlain yellow perch, and then identify them. So are the parasites in Lake Champlain yellow perch, is it the same parasite that's in the Great Lakes system? Maybe so, maybe not. And then we wanted to describe pathology infection. So we teamed up with Inga Sidor, the veterinary pathologist. And then we also we did some preliminary investigations looking at infection with fish health. So fish sampling, we actually didn't go out and kill fish for our study. This was great. We received fish that were already being collected as part of other surveys or studies and were bycatch. And so contributors, we had Bernie Pianka at Vermont Fish and Wildlife, Ellen Marsden at UVM, and postdoc and grad students. We have private anglers up on the island. Mark Allard is an angler in Keeler Bay. And then Ray's Seafood Market has also contributed a number of infected fish, really heavily infected fish, actually. And so for fish sampling so far for our study, we've screened 400 fish. We have way more in freezers here at UVM. We just haven't been, don't have the resources yet to go in and sample those. Again, all fish were received as bycatch. We have 6 sampling sites. Our goal was to sample up and down Lake Champlain, and sampling methods were angling, electrofishing, gillnet, and trawling. And so all fish were measured and they were weighed, and by using these 2 measurements together, we could come up with an index of body condition for each fish. And then for sampling fish for the heterosporous parasites, we filleted each fish and did visual examination. And then we collected a small fragment of tissue from the same area and put it on the wet mount on a microscope side to look at the parasites under light microscope. And then we also put samples away in tubes for PCR screening to screen for the parasite DNA from the fish sampled. And then in addition, heavily infected fish were embedded in formalin for histopathology analysis by our veterinary pathologists at UNH. And so here are some pictures of infected fish. KB is Keeler Bay. You all may be familiar with Keeler Bay up on the islands. And as soon as you fillet a fish, you see these opaque freezer burned sections of the skeletal muscle tissue indicating positive infections. So wet mount light microscopy, we put these tissue sections under the microscope and we can actually see the parasites themselves. They're pretty fantastic. You have spores, sporoblasts, and then these larger sporophore cyst vesicles that kind of, in an infected fish, they overcome the skeletal muscle tissue. For methods for PCR-based screening and identification, this is similar to, say, PCR-based screening for SARS-CoV-2, or so we basically extract DNA from our tissue, and then we screen specifically for the parasite DNA. And then in positive infections, we amplify the infection by DNA sequencing. And then we use that DNA sequencing, the sequence of A, C, T's, and G's to identify the parasites, and then compare it to other heterosporous parasites that have also been sequenced. And then also to understand the relationship of the parasites from our Lake Champlain yellow perch to other heterosporous parasites. Okay, so by histopathology, these are results of tissue that was embedded in formalin and then sent to UNH, the diagnostic veterinary lab, where they did cross sections and embedded it in stain. And what I want you to see is, and looking at all these images, for me, not being a veterinary pathologist, it's hard to say, like, what are we looking at? But basically what I put next to results in histopathology, that is that pink rectangle, that's what normal skeletal tissue looks like. And the images above, the larger ones, is what the infected tissue looks like in our infected fish. So instead of this pink tissue, you see the invasion of all this other non-pink tissue, which is the heterosporous parasites. So they basically take over the skeletal tissue, liquefying it. And it's because the parasites produce millions of spores. And so over time, it gradually completely destroys the muscle cells and tissue. So here are some other pictures of the histopathology with different stains of the parasites in the muscle tissue. And you can see that these tissues are primarily all parasites and spores. In terms of the distribution of infection, but just by visual examination of the fillets, we found infection in Missisquoi Bay. 3 out of 80 fish were infected. In the gut, 2 out of 81 fish were found infected. In Keeler Bay, 2 out of 71 samples were infected. We didn't find by visual examination of fillets any fish infected on the middle of the lake over in New York in Corlear Bay, or on the Vermont side in Burlington Bay. And then we had a limited sampling down from Larrabee's Point. And so Southern Lake Champlain, and we didn't find any infection by visual examination of fillets. And we haven't gotten around to doing the PCR-based infection, meaning that from the same fish that we examined the fillets visually, we also do PCR. And other studies that have done the PCR and visual examination have found that you have way more infected fish than you see the parasites in. So in previous studies in the Great Lakes region, only 20% of the PCR positive fish actually had the visual infection in the skeletal tissue. So we're likely grossly have an underestimate of the true prevalence of this parasite. And so PCR screening is next on our list of things to do. And then this also brings up a question. So, if only 1/5 of infected fish show signs of infection in the skeletal tissue, why do some show signs of disease and some don't? And so that's a question we're interested in. And so what are the parasites that we're seeing in Lake Champlain yellow perch? So by histopathology, they are similar to Heterosporus sutherlandi, which was described from yellow perch in the Great Lakes. So it seems that we have the same exact species of Heterosporus here in Lake Champlain, indicating that invasive Heterosporus parasite in the Great Lakes region has definitively spread here to Lake Champlain yellow perch. And then by genetic work, we also found that if we compare the DNA sequence from our Heterosporus parasites in Lake Champlain yellow perch, they're identical to the parasites from the Great Lakes yellow perch. And then also by phylogenetic analysis, looking at closest relatives by genetic analysis, we also found the same result. So it seems that the Great Lakes Heterosporus parasite has made its way here to Lake Champlain and is now the definitive parasite infecting our Lake Champlain yellow perch. So then we looked at body condition by measurements of body length and mass for fish from 3 bays, Missisquoi Bay, Keeler Bay, and Larrabee's Point. And the solid line on these graphs shows the standard weight equation if you look at weight and length together. And so that's kind of the normal or average body condition for yellow perch. And so from Larrabee's Point, you can see that all the dots are individual fish that we measured and weighed from Larrabee's Point. They kind of sit on that standard weight equation, showing that weight and length are standard. But if you look at the fish measured from Keeler Bay and Missisquoi Bay, you can see that our data points sit below that standard weight equation, indicating that they are of lower body condition. Interestingly, we didn't find infection in Larrabee's Point fish, but we did find a Heterosporus infection in Keeler Bay and Missisquoi Bay, which may be correlated with poor body condition. We don't know. This is something that we're interested in looking more into. So conclusions from our study is that Heterosporus infection was detected by visual examination of yellow perch in 3 bays in northern Lake Champlain. That's Missisquoi Bay, Keeler Bay, and the gut. Heterosporus infection causes significant pathology in infected fish in this study. The Heterosporus infections in Lake Champlain are definitely Heterosporus sutherlandi, species described from Great Lakes region. And the fish infected in northern Lake Champlain have poor body condition. So there may be some correlation between infection and body condition. So further study, we have a lot of further study to go. Our results are very preliminary. So we hope to do PCR-based screening to see the true prevalence of this parasite. So by visual examination, we're likely getting a very gross underestimate of the true prevalence and also distribution of Heterosporus in Lake Champlain yellow perch. We'd also like to survey other fish species in Lake Champlain because the studies in the Great Lakes have shown that a diversity of other fish species also harbor infection by Heterosporus parasites. We'd also like to look at heterosporous infection and to see if you find it in co-infections with other parasites and pathogens. So yellow perch are infected with a wide diversity of other parasites. Are they more likely to be found infected with heterosporus if they have other infection? That's a question we'd like to look into. We'd like to further look at fish health, infectious status and intensity. And then also, why are some fish infected and some aren't? And so look at role of contaminations in infection status. So we know that heavy metals like mercury and lead can decrease immunofunctioning in fish, as well as exposure to various contaminants, including PFAS and various PFAS compounds. And so something we'd like to look at is, we have all the fish that we've taken muscle samples from. Do we see differences in contaminant exposure between infected and non-infected fish? And then also going forward to better understand the distribution of heterosporous parasites in Lake Champlain fish. Is there a way to screen fish for infection without having to actually take the fish itself? So other non-lethal ways to determine infection. So what is the threat of heterosporous parasites? This is a big question we get. We don't know. So in Minnesota, where it's been established for longer, a modeling study published in 2018 indicated that a 10% increased mortality rate from heterosporous infection can lead to a 30% reduction in yellow perch yield, as well as a 10% reduction in mean catch weight, which makes sense. If you're a fish and you're infected, you're not eating as much, your body's putting energy into combating an infection, you're going to be not as big and not as healthy. The Great Lakes Fish Health Committee has listed heterosporus as a restricted pathogen, and heterosporus species are also listed as reportable pathogens or a disease of concern in many different states, including Illinois, Maine, Michigan, Minnesota, Utah, and Wisconsin. So in states such as Maine, where heterosporus parasites have not been documented yet, it's definitely something that they keep an eye out for every year. And then something also of consideration here in Vermont is we found it in yellow perch in Lake Champlain. We haven't been looking for it so much in other species, and we actually, it hasn't been documented in any other water bodies in the state yet. And so that's something else to consider, is the spread of this into yellow perch populations in other water bodies in our state. What is the threat of heterosporus? For me, it's been really helpful, but also illuminating to talk to anglers. And so, you can hit up ice fishermen on the lake in the winter and talk to them. And so many people have seen, have filleted their yellow perch to pan fry them, and right away they see the parasites. And so they're well familiar with these parasites, are well familiar with infected fish. The state does get a lot of concern, the fish biologists, because of heterosporus, because it's so visible. And a big question they get is, can I eat infected fish? Is it okay? But I've been in contact with a number of anglers and folks who make their living doing fishing tours on the lake, including Paul Dunklin, president of Ray's Seafood Market, who's so knowledgeable about the health of fish and has been out on the lake taking anglers on tours, as well as selling fish through Ray's Seafood Market. Ray, Paul Dunklin, who's the owner of Ray's Seafood Market, has documented heterosporus in yellow perch for about 10 years and has indicated that it's getting progressively worse. And to him, it seems to be spreading through the whole lake now, which is something that we didn't see through our sampling. But again, we have very limited sampling for our study. Again, our study is pretty preliminary. And so, according to Paul, I believe he quotes, "I believe we should learn everything we can about this pathogen because it's here to stay. We know we will have to live with it, but knowing its effect on all fish is important." So what it seems that we have in our preliminary data suggests is that this is an invasive parasite that's shown up and is emerging in Lake Champlain yellow perch. But it seems that it's endemic now. And as Paul says, it's here to stay. And so this really brings up the issue of emerging infectious disease. And so it seems we have this disease in yellow perch. And again, it's going to take a lot more work and research to really understand what's going into the disease, what's causing it. It's not just a novel invasive pathogen, but what's going on with the host? Why do we have it in some bays and we don't have it in other bays? Why are some fish showing signs of infection and some aren't? But also the environment. So there's a lot more work to do on this project, but I'm just happy to share our preliminary results with you today. What can we do to control spread of heterosporous parasites in Vermont? We can make sure that we do not move live fish from one water body to another. So again, while we've found heterosporous in Lake Champlain yellow perch, we haven't found it in yellow perch in other water bodies in the state, which is good news. We want to make sure that infected fish are not discarded back into the water, but they're placed in the garbage. And then also make sure not to move water and potential microscopic invasive species from one water body to the next. So because Heterosporus is a microsporidium with all these spores, you can move infection just from moving water. And then also clean, disinfect, and dry all equipment. So the same precautions for moving other invasive species, both fish themselves and pathogens all apply to keeping Heterosporus contained within Lake Champlain. So acknowledgements, there's a number of different people who have contributed fish, but also Emma Privet, who was an undergrad here at UVM. A lot of this work was done for her senior research project in my lab. And then funding was through UVM. And I'll end it there. Hopefully that all went through.
Yes.
Great.
Thank you. And maybe, Ellen, if you want to stop sharing, then we can look at each other for questions.
Okay, let's see.
Unless people have slide-specific questions, we can pull it back up.
Yeah.
Thank you so much. I'll open it up for questions. Go ahead, Brett.
Yeah, Ellen, really worrisome but excellent presentation. Thank you. You touched on this, but can you state whether it's known whether there's a human threat to consumption of diseased fish?
Yeah, that's a good question. That's probably like the most common question we get. And so we cannot get infection by eating infected fish. Yeah.
Okay, thanks.
Yeah. Andrew.
I echo, thank you for the presentation. It's very informative and also disturbing. It seems very likely that this parasite came into Lake Champlain via the Great Lakes through one of the ways that fish and other organisms get from the Great Lakes to Lake Champlain. Any idea where it came from before it got to the Great Lakes? Where is it endemic to? Is it from Europe or Asia, or any knowledge about that?
That's a really good question. So it's unknown. There's a few different hypotheses. One of them being that it may have come from aquarium fish that were released into the Great Lakes. It hadn't been documented in North America before, but it's more common in different parts of the world, including Southeast Asia. And so I think I listed some of the fish is found, and it's also found in eels, and it's found in aquaculture, but mostly on the other side of the world. So it seems that it was likely introduced. Although there are a diversity of microsporidian species that we have here in North America, that was the first documentation of the genus Heterosporus in North America. So it's thought that it may be from release of ornamental fish.
Thank you.
Yeah, hi. Excuse me, this is Bob Fisher and Barry. Hope you can hear me okay.
Mm-hmm.
I'm getting some feedback. So Wow, this was a very uplifting thing. I know about round Goby and everything else. But as a biologist, former fishery biologist, and in charge of large wastewater and water systems, I'm used to cryptosporidium and whatnot. And I know how difficult it kills scores. And this is completely off the wall, off the top of my head. But I'd ask you to research throwing it in the trash. Um, I can't say that by the time it goes to a landfill and then goes to but it's the same as pharmaceuticals. It's probably not the same, but eventually that leachate is going to come back to wastewater plants. If it goes to the Montpelier plant, there's a small possibility that it could then infect their areas. So I myself would just throw them on the ground. And I don't want to be quoted for that, but nonetheless, that's just off the top of my head with this limited knowledge on a different cross thing. Thank you.
That's a really good point. Yeah, I didn't even think about, put it in the trash, but then it's likely to go back into the system. I know that in the experimental studies that they've done in Minnesota, that they've found that it can persist in water for months and months. So it's a pretty stable parasite. So yeah, I'd be curious to know if it could go through the system. Yeah, that's a good point. Has anybody on this committee seen infection in a yellow perch?
No.
No. Okay.
I have a quick question of the CAC, actually. We had a list of all of the individual species that make up the invasive species that have shown up in Lake Champlain. Are these on that list? Are these pathogens on that list? If not, we should update that list.
Yeah, I don't think I don't think they are.
Yeah, I don't believe so.
I was just wondering that myself, Breck. So that's a really great conversation. And I wonder also about Lake Champlain Basin Program, just whether they've considered that.
Yeah,
it may not be on the list yet because maybe because we hadn't identified it yet. So we knew we had heterosporous parasites, but the big question was, is it the same parasite that's in the Great Lakes ecosystem? So maybe now that we've identified it as heterosporous sutherlandi, we know what species it is. It can be promoted to invasive, the invasive species list.
Ellen, you started your presentation. There's some feedback here. I don't know where it's coming from, but it seems to be hitting all of us. You started off your presentation saying that there were several different pathogens that had shown up in fish since the early 2000s in Lake Champlain. Could you send that list to Karina? And we can work with the Basin program to see about making sure that those are included.
Okay.
And I see Paul has a question.
I do. Ellen, great presentation. Thanks for sharing.
Yeah.
You had, and I know the study is primarily on the yellow perch, but you have mentioned other species. Do we know to any degree how it's impacted those other species that you mentioned?
Yeah, so so far it's only been visually documented in walleye in Lake Champlain. We don't have any documentation from the Vermont fish biologists of anybody seeing it in other species, not to say that it's not there. In the Great Lakes, they've done a lot more PCR screening of different fish species, and that's how they've founded the infection. So we haven't done that yet. We haven't done PCR screening of different fish species in Lake Champlain. So that's definitely something that needs to be done. I mean, so we know we know it's circulating around in bays in northern Lake Champlain, and anglers have documented it in additional bays in Lake Champlain that we haven't sampled yet. So, given how prolific these parasites are, and that they're circulating around in the water column, it's all the other fish species must be exposed to this parasite. So if they're are they infected? Is a really good question. So that's something we'd like to further do, is to broaden our survey to other species.
Yeah, and even to the birds and other that feed off of the fish, and the impact that it's having across that population.
Yeah, because we know a lot of birds love yellow perch, as much as humans do, right? Like common loons love yellow perch, great blue herons. So yeah, it would be really interesting. I've thought about that. So, my background is in bird parasites. So I'm always thinking of the birds, but and I actually have a project on common loons, not on Lake Champlain, but across New England. So yeah, it would be really interesting. We would just probably need, it was difficult to sample birds, especially like lake birds, but I'm wondering if even just fecal samples may suffice for PCR-based screening for these parasites. That may just show that the parasites are passing through them, but if it's infecting bird species is something I don't think anybody's looked into yet. So, yeah.
Right, and the migration of those birds is of greater concern. Now it's not just in the lake bodies; it's everywhere those birds go.
True, and that may through the bird host may be how it got here from the Great Lakes too.
That's a good point.
And so, it'll be really interesting to track this parasite and see how far it spreads. I've talked to Maine fish biologists, and they definitely have it on their radar. And so, if it was first documented in the Great Lakes in 2010, in 2000, and then we found it here in 2014. It's just a matter of time before it keeps moving. Yeah.
And yeah, hi, this is Bob Fisher again, just real quickly. Although I think that last question clearly eclipses mine. Another reason for boat washes and whatnot. Obviously, they have the fish, they cut it up, they chop it up, they don't want it, whatever. Say they dispose of it properly, but it still ends up in their bilge water from the washing down and whatnot. They go somewhere else and spreads it. Although obviously, avian spread could clearly be much more important. Thank you.
And I just wanted to make sure I heard this, but is yellow perch the only commercially harvested fish in Lake Champlain? Is that correct?
I think now it is. Some of you may know better than I do. I'm not a fish biologist, but I think that's the case now. Yeah.
And the seafood market can still take this fish and serve it to humans, or they're not taking the fish?
And so what they do at the seafood market is that, an angler will catch like a bag of yellow perch any time of year, bring it in, it goes into the market, it's bought by pound. And then what they do in the back is they fillet the fish before they put it out for sale. And it's upon filleting that they see the infections. So, and so it's for that reason, it's been really helpful to talk to Paul, who's like doing, and others who work at Ray Seafood who are doing the filleting, and the impacts of this disease, really play out at that level because they're trying to sell the fish. And so what they do actually now is that they'll do like a quick incision and look in before buying fish just to make sure that it's not infected, especially during the winter season when they see an increase in the prevalence of this parasite in yellow perch.
Thank you.
Yeah.
Just one other quick question. You said that this is transmitted in live fish. There's no chance of contamination within something like raised seafood and it being transmitted to other meat, that is in the store.
Could it be transmitted through the store?
Yeah, is there a danger of the premises being contaminated, or is that not? That's impossible.
Oh, that's a good question. And I'm so curious. Like I've talked to Paul Dunklin so many times. I've actually never been to Raise Seafood, but I think there's multiple locations. Is that correct? Does anybody know? Has anybody been to Raise Seafood or bought fish from Raise Seafood? But I think is there more than one location?
There used to be another one on North Avenue. I'm not sure whether it's still open. I think the only one that's really operating is in Essex.
Okay.
But they also have a seafood truck that travels and sells out of the
Okay,
truck.
didn't know that.
But that's a good question. Any other questions for Ellen? Before we move on to our next, that was really, I think, enlightening for the committee. Thank you so much.
Well, thanks for inviting me.
And Ellen, if possible, if you could send your slide deck as a PDF and share that, then I can share it with the committee. That would be very helpful as a reference.
Okay, great. Thanks everybody for listening.
Thank you so much.
Thank you.
Thank you.
And I'm just wondering if I still see Ellen's slides on the screen, so I'm wondering, Ellen, if you can stop sharing. Oh, there, perfect. And we can transition to Hillary. Hillary, has your son come home yet?
Yes, he made it home safely.
Oh,
Yay.
good.
Let's see. Yeah, I'm just going to share my slides. Oh, hang on. Um, it always hides the there we go. Okay, how's that look?
That looks good.
Okay, and how's the feedback? Are you guys hearing me without that feedback?
Yes, no
Sounds
feedback.
good.
Okay, perfect. Okay, so I was asked to cover the clean water service providers and give an update on sort of where the clean water service providers are right now, how the program's evolved a little bit, maybe what the program is. And we'll just use the South Lake Clean Water Service Provider as our case study since we are a partner with the Rutland Regional Planning Commission in administering the South Lake QUIS. So I've got a couple slides here that are from the state of Vermont that just kind of basically discuss how the QUISPs fit into the rest of the funding, programming, regulatory, non-regulatory water quality world. So the QUISPs are in that, so everybody, of course, is familiar here with the TMDL for phosphorus for Lake Champlain, which is the driver of the clean water funds and the clean water service providers. So I'm assuming everyone's familiar with that. The TMDL looks at both regulatory reductions in phosphorus through ANR and Agency of Ag and the VTrans and other regulatory programs who issue permits and have programs in place that regulate sources of phosphorus. And then it has a bucket of phosphorus that the Clean Water Fund would help reduce. That's non-regulatory. So looking at working with towns, working with landowners on non-regulatory volunteer projects. And that's the pot of funds that's sort of the place where the QUISPs sit is in that non-regulatory project area space. And then the TMDL sort of, through the compilation of all those project types would hope to meet their phosphorus load reduction targets. And here is you've probably familiar with this graphic and looking at the different sectors. So agriculture, developed lands, stormwater, roads, and then there's the forest sector as well, which falls under natural resources. So streams, forests under natural resources, lake shorelines, and then wastewater, which some of us are more familiar with than others, but which is the other sector that is counted in the TMDL. And each of those sectors, there are requirements for reducing phosphorus through each of those sectors. And the CLIPs are working in the natural resources sectors, the developed land sector, and a little bit in agriculture, and we'll go into that in a second. So these are the sort of regulatory versus non-regulatory reductions. And for agriculture, for example, Agency of Ag is in charge of the funds and the grant programs and the both regulatory and non-regulatory sort of reduction of phosphorus in that sector. And then there's a little bit left over, this 10%. It's in the non-regulatory side, and that is the part that the QUIS, those would be the project areas that the QUIS would work in. So for example, the QUIS wouldn't fund a manure storage pit, but they could fund a planting, like a buffer planting on a stream that's on a farm. And then similarly, the other one to look at, I think, in this list is the forest sector, where remember the gap watersheds, where the modeling for the TMDL they didn't even applying all of the best management practices across certain subbasins, they couldn't, like all of the phosphorus couldn't be accounted for through those best management practices. So the leftover phosphorus, or that gap in the phosphorus that could be accounted for, was put in the forest sector. So for Basin 2-4, which is our basin, South Lake, and for Basin 6, which is Missisquoi, there's the extra phosphorus got put in the forest sector. And we're going to see that that's going to come back in a second, because the CLIPs are funded through a formula, and that forest sector piece comes into that formula. So keep an eye out for that. So the clean water service, these are the list of the clean water service providers. There's the Vermont Housing Conservation Board, and then a number of regional planning commissions. And then for South Lake, it's the Rutland Regional Planning Commission partnered with Poultney-Meadowy Natural Resources Conservation District. Yeah, so that's the distribution. Right now, the clean water service providers are just in the Lake Champlain Basin. So the partnership with the Poultney-Meadowy and the Rutland Regional Planning Commission, we felt would be advantageous, because the RPCs and conservation districts are both statewide organizations, and we're set up in statute similarly. Poultney-Meadowy does a lot of project identification, project development, implementation. We've got a history of doing assessments on the landscape, so we've collected a lot of projects already sort of prepared to go into this funding source. And then the Rutland Regional Planning Commission has they have multiple financial staff, in-house mapping, and then like nice data storage systems sort of already in place. And we'd worked together in the past. So just kind of looking at the South Lake Quest, that's how we ended up partnered to do that work. And then this was kind of a new funding delivery mechanism. It was a change from A&R managing all of the funding. All of the applications would come in the past to A&R, for example, through the their old they had a couple of the Clean and Clear or the Ecosystem Restoration Program or some of their grant programs. So now these applications for projects are coming in regionally. And so the idea being that it would be more efficient, and that the regional groups would know the area, know the projects better. The Basin Water Quality Councils are made up of a number of partners, and we'll go into the makeup of the BWICs. But they have the oversight of whether a project is accepted or not, whether it's funded or not, and then the QUISP sort of manages the administrative part of funding that project. And we do a lot of I'll go into a little more detail in a second. So these are basically the sort of responsibilities of each of the groups. And so A&R established the QUISPs, created the formula that ties back to the TMDL in terms of the cost efficiencies and the targets that we needed for phosphorus reduction. And they provide a lot of technical assistance to the QUISPs, and then also evaluate the QUISPs that they're making satisfactory progress toward their goals. The QUISPs follow they have their own contracts, these water quality restoration formula grants, which are based on a cost rate methodology, and I'll go into that in a little bit too. But basically, they set the cost efficiencies that we need to meet for our phosphorus reduction and the price of the projects that we're paying for. We also facilitate those basin water quality councils, which are a guiding council for each of the QUISPs, made up of has a specific membership. We sort of facilitate or oversee all phases of project completion, including from identification to implementation through these funds, just for the non-regulatory projects, and then develop partnerships and run grant rounds. And then one of the new and exciting things that the QUISPs are able to provide through state funding is ongoing operations and maintenance for all of the projects that we fund, and also for a select number of projects that we decide to adopt. So that would be paying for like ongoing maintenance over time, which is a new thing and is exciting. And then we report our progress to DEC. So this is the South Lake QUISP area. The Poultney and Meadowy watersheds line up surprisingly well with the Poultney Meadowy watershed, the conservation district boundaries. So that was nice. And then the Southern Lake Champlain Direct, Southern Lake Champlain Direct is that purple watershed that goes up, borders Lake Champlain going north into Addison County. So we have the coverage area for the South Lake QUISP is not just the Rutland RPC and Poultney Meadowy, it also includes Addison County RPC and Otter Creek NRCD. So we work together in those areas where we have shared projects, or they've got more time and experience up there than we do. So we're kind of working up in their area for that sub-watershed. So the basin water quality councils that I mentioned that oversee selection of projects for funding are made up of 2 members of regional planning commissions, often the board members. Occasionally it's a staff member, 2 conservation district representatives, 2 watershed group representatives, a land conservation group representative, and then 2 representatives from municipalities. So they're pretty diverse, and we have a great Basin Water Quality Council that provides great input and is really, really nice to work with. Sorry. Sometimes my computer doesn't like to. There we go. So I kind of went over this already. We have limited watershed organizations in our area, so we were able to have the Nature Conservancy and Vermont Land Trust count as the Nature Conservancy and Trout Unlimited are our watershed groups, and then Vermont Land Trust is our land conservation organization. But we don't have any watershed groups like Friends of Northern Lake Champlain type of watershed group. So then sources of projects. So we have, because of the fact that we don't have those other watershed groups in the area, we have limited capacity for applying to our funds and running projects. So we end up, the Poultney Meadowy Conservation District was the sort of main implementer in this watershed. So we do run a lot of projects, and the sources of those projects include things like stormwater master plans, geomorphic assessments, any kind of scoping study that's gathering a number of projects from the landscape. And then we get calls too from landowners and towns who have just drainage and runoff concerns, and those often end up being more likely to be a project because we have landowner support or town support going into it. Like, they're already ready to solve a problem. So we've had trouble with the stormwater master plans or geomorphic assessments because then we're approaching people saying, we think you have a problem, would you like to solve it? So that's been a challenge for us as we try to build our projects and our applications for the funding source. So we work under the QUIP funding policy, which governs all of the clean water funds for the state of Vermont. And the QUIP funding policy divides projects into these categories. So they're divided into the different sectors, which we talked about a little bit already. different project types, and then also different project phases. And we'll talk about this a little bit more too. But there's a lot of specificity and a lot of detail in the CWIP funding policy, and so our projects need to meet those specifics from the CWIP funding policy, which can be challenging. Oops. So every quarter we run a grant round. We try and encourage people to turn in applications whenever they're ready, and often the QUISP staff works with folks. Like a lot of times there are applications, or we help people write applications. We really want people to apply to the funds and to manage projects in our area. So we try to help them as much as we can. We have a pretty simple application process, but there's a lot of vetting that happens. So we have a pretty simple application, but a lot of vetting that happens with our partners at DEC. So multiple folks at DEC will review every project before it even comes into a grant round. So it's a pretty collaborative process. Then the BWIC meets, so the Basin Water Quality Council, or the BWIC, meets quarterly to vote on projects. The QUISP staff reviews the applications, and we provide a score for the project. And the largest part of the score, half of the points for the score, is that cost efficiency by sector. And we talked about that a few minutes ago, that everything's kind of divided up. And I'll show you some tables in a second that shows you the sort of cost per sector that we have to work with. And the cost rate methodology was just updated by DEC. So we used to have to try and meet $9,000 per kilogram of phosphorus reduced. That was our cost efficiency, but it's doubled now, and it's 21,600 per kilogram. And I think people were just really having trouble making that formula work. And prices increased quite a bit over the last few years, and for a variety of reasons, the formulas got updated. So we can look at that in a second. But we look at cost efficiency. That's the most significant factor we look at, and then the life expectancy of the project, the complexity of the ongoing operations and maintenance, since we'll be maintaining that project over time, the value of the project within larger goals of the basin, like is it a high priority project for multiple reasons, and then we look at co-benefits. So these would be things like habitat or community involvement. And we add points for co-benefits, which is 20 out of the 100 points is co-benefits. That's the second biggest points that we add. And then once the projects are scored, they go to the BWIC, and the BWIC votes them up or down, yes or no, or they can vote, they might have a contingency, but they approve the project. So these are the revised targets that I was telling you about. So the original targets are in the second column of numbers there, and the revised ones are in the first column. And you can see that the overall change is 49% when you add everything up between all the different QUISPs. So that has definitely helped us toward meeting our goal. We were pretty far behind until this happened. And then on top of being behind, when this happened, we've recently completed a bunch of projects, so now we're less behind. So we're feeling a little more optimistic. So these are the different sectors and the kilograms that we're supposed to try to reduce per year per sector. So our total's roughly 30 kilograms per year. And you can see that we are one of two basins that has kilograms required to be reduced in the forest sector. So those are a little challenging because the modeling didn't show that it came from the forest sector, but extra kilograms got put in the forest sector. And there's pretty limited project types that we can use in that sector. So right now we aren't holding ourselves to that formula. Like we aren't saying that half our kilograms have to be in the forest sector. We're just taking projects as they come and trying to meet the total reduction per year. But that will be something that would be a challenge if we were held to that formula. So this is kind of the inner workings of the formula grant. These are the numbers that support the funding. And then these are examples of projects for those different sectors that we are able to fund. So for project development, we can support like a broad range of project identification type studies like stormwater master plans, watershed action plans, geomorphic assessments. For agriculture, it's a little trickier. We only can support natural resource projects on farms. So that would include like a buffer planting on a farm, for example, as I mentioned before. or a stream, like a stream restoration, a floodplain reconnection, but not a true agricultural project. So it's a little hard to tell sometimes if it's an ag project or if it's a stream project. And those funding levels are significantly different between the two. The cost efficiency levels are different. So we're working through some of that now. And then for developed lands, we do have a lot of flexibility in that category. We have very little kilograms in our formula for developed land, so not a lot of funding at that height. That's the most expensive category. So the most dollars per kilogram. And because we have so little developed lands, our formula is actually like has the least dollars per kilogram. I think we're the least or second least dollars per kilogram in our formula because we don't have these developed lands. But we have a lot of developed lands projects that we've identified and are trying to fund. So it's sort of skews us toward being expensive. And then forest roads is one of the options, but the credits aren't very high for that project type. So it's not very efficient. And then streams, floodplain reconnection, dam removals, and river plantings are examples of project types. So operations and maintenance, this is a definite, I think, bright spot for this funding. It's exciting. It makes it easier to work with private landowners and even municipalities when they know that they're going to get some help with any maintenance, ongoing maintenance for the project. They also just know that they're going to have technical support over the years because there's a schedule. And after the project's installed, every so many years, we go out and verify that the project's still working and would bring information back to the QUISP if it wasn't, to help make sure that that project continues to work. So I think that's something exciting that DEC did. And I look forward to, in fact, we have our first, our stormwater, we had a big stormwater project in Fairhaven, and the vac truck is coming and it's going to clean it out on Wednesday. And the town inspected it and called A1 to come and clean it out, and we're going to pay for it. So it's a big project. It treats 24 acres of stormwater. And then just looking at some successes we've had locally, we have a grant that the WIC approved that's kind of just a pot of funds that's for some quick engineering visits. So if we have landowners who call us about projects and we're not quite sure if they would be best going through the QUISP or another funding source, we just have a little bit of funds where we can pay somebody to come visit the site, look at it, make us some maps, and then we will make a recommendation about whether that goes to the QUISP or enhancement or, another funder. So that's been really helpful. And 13 projects have been through that program and then either moved on to other funders. Sometimes other organizations have managed that through other funds, sometimes to the QUISP. But it's sort of a way to keep things moving and get projects that aren't part of a study, a landscape scale study, into the program. And then adoption too, we get to adopt projects. So if we have projects that are great projects that are in the community that don't really have a responsible person for them, or their contract is already, run out, then we can adopt them and pay for the maintenance there. So we adopted the rain gardens behind the Pulteney High School, a buffer planting on a farm that's a really nice 50-foot buffer, and then a stormwater project on Lake St. Catherine. Those are the first three that we've adopted. And then these are kind of our projects funded over the last couple of years. And you can see the project numbers aren't very high. And we've funded most of the projects that have come to us, unless there was something, significantly wrong with it. So we would love it if there were more partners in our area and more people to bring us projects. You can see, though, the orange bars in the chart are our project completion, and we did have quite a few projects finish up over the last couple of months. So we're hopeful that, the sort of, now that we're used to this work, and hopefully have more people working with us. Like, we've been cultivating relationships with lake associations. We have 2 lake associations who are running projects. We have a couple towns running projects, and continuing to build those relationships and encourage people to apply to the funds is helping. But part of the work of the QUISP has been, growing those relationships and helping build capacity locally as well. So we do some trainings with folks. We have like an invoice SOP, so people know what information to put on invoices. Like, we've been trying to do as much of that capacity building locally as we can. And then it's a complicated program. So it's, you saw all the sort of charts and tables, and there's a huge, like if this, then that table that goes with the equip funding policy that shows whether or not a project's fundable. So it can be daunting, which is why we work with our partners so closely. And I just have examples of projects that we installed in the past that are like road runoff draining through a barn and then to a gully. And that used to be fundable. And now I don't know how we could fund that because it would be like 3 different agencies involved. And things have just gotten a little more siloed and a little more complicated maybe with the TMDL work. And then sometimes I hope DEC, sometimes I would wish that DEC would be a little braver and let us do things and then figure out the crediting as they go and apply that back to our projects if they think it's a good project type. So we're waiting for some projects like stream gullies, which they don't love to fund maybe, or strategic wood addition. We're waiting for that methodology to be worked out so we can use those project types. And then let's see. Yeah, I think I've already been over this a little bit, just that the project types are limited. The formula grant's pretty specific. And then staff capacity, I talked about that. Our capacity is really low in our area, just without other watershed groups to absorb some of this work. And then I also have felt that the QUISPs get a lot of capacity building through the administrative funds. So 15% of the formula grant goes to the QUISP for administering the program, which is capacity building, but there's not that volume of funding for other project managers or implementers. So it doesn't help stabilize or grow the watershed groups in the area that are going to implement these projects. So that's going to create a situation where the QUISPs feeling pressure to get the funds spent we'll hire staff to implement projects, which is something as a conservation district staff person that I worry about a little bit. And then just ending on some positive notes, the South Lake BWIC is amazing. It's a great group of people. They're really engaged. They're really working hard to make projects work and to come up with projects themselves. One of our BWIC members is actually working on the strategic wood addition crediting with DEC. So it's a really great group of folks to work with. DEC has been really good. They're always really responsive. They call back right away and answer questions and figure stuff out because it feels like every situation, this is all complicated and we come across new situations every day. So we call, well, a large number of them often, and they're always great. And then just it feels like the pace of project funding and development is increasing over time. So hopefully that will continue. And I think that the desire to grow capacity for project implementation is consistent across partners. I know DEC knows it's an issue, LCBP knows it's an issue, and those groups have put out some organizational support funds to help increase staffing at those groups. And then DEC has a whole action list, like tier 1, tier 2 actions that include a lot of the things I've mentioned. And so they are working on, adding more project types and some of the things I just talked about tonight. And that is it. I don't know if anyone has questions. I was worried I was going to run really long, so I'm glad to see that now that I can see my clock, it's not too late.
No, that was really great. Thank you so much, Hillary. Open it up for questions.
Hey, Hillary, that was fabulous. So I have not been paying as much attention to this as I ought to have been, and this just is really informative. I have a couple of questions. The first one is, this one's fairly down in the weeds, but how do you calculate the cost efficiency for a project development sort of project? And maybe stated slightly differently, those are things that don't create an immediate reduction in phosphorus, right? So how do you prioritize it against those projects that do have a demonstrated reduction in phosphorus?
Yeah, so basically we can't. So the formula grant includes a percentage that's for project development and project identification. So we just use those funds. And to be honest, because that's such an issue in our area, anybody who's wanted to do that work, we have said yes to at these in these early years. And one of the things I forgot to talk about is that we did a lot of project development work. Holy Meadow, we did a lot of project development work through geomorphic assessments and stormwater master plans, and we identified a lot of projects, but only a small percentage actually meet that cost efficiency requirement. So it is pretty stringent, and it's loosened up now, as you saw. But in the beginning, it was really stringent. Like I think 3 out of my 200 projects actually met the requirements, and now it's increased, which is good.
Super, thank you. I have a bigger
Yeah.
question, but I'll defer to Andrew and Bob.
I wasn't paying attention to who was next, but I see Bob.
Oh, real quickly. No, I'd actually, his question's probably much better. I just, yeah, excellent presentation. Thank you. I just wanted to applaud you. It's not a question at all on overall on O&M, because that's what I often see, and as an infrastructure type person is, these wonderful things, but then the O&M's difficult. And if there's no funding for that, that's extremely difficult. Thank you.
Yeah. Thanks. It's true. And Andrew.
Thank you for the presentation and the great work you're doing in the South Lake. Really important. And I'm just thinking about you talked about co-benefits a little bit, and wondering whether thinking about the co-benefits and other types of work happening in the South Lake, whether that's a way to get more partners involved, as someone that's really coming into this for restoration of fish and wildlife habitat, yet that's going to have really good benefits for phosphorus reduction. Are you able to seek out partners that are active in that or other types of work that you could through the co-benefits get to the phosphorus reduction? I don't know if that made sense, but
Yeah. So if the project was mainly not a phosphorus reduction project, it was mainly, say, a habitat project, then it would probably not be a great fit for the formula grant, but it would be a good fit for an enhancement grant, which is sort of the other side. Like the Clean Water Fund has 2 grant sources, the enhancement grant program and then the formula grant program. But I think that's what you bring up, is that, our main partners in the area who do work there with us are the Nature Conservancy and Trout Unlimited. And they're very interested in stream restoration and habitat. And so that's where strategic wood addition is something that's really hopeful for us. And so we're hoping that the crediting doesn't become so burdensome that those projects end up in a different funding source. We're hoping that the crediting is just based on the fact that it's improving processes in the stream, which would reduce sediment, which would hold sediment up in the headwaters instead of letting it pass through. And so hoping that goes smoothly, because I think that's where our partners have participated, or the lake associations have worked on LWAPs and some stormwater projects around the lakes.
great. Thank you.
And Breck, you had another question.
Yeah, so I'll ask, I guess, my higher level question of you, Hillary. You're in a unique position of being on the CAC and deeply involved in this CWIS. I'll ask you the question that we ask all of our presenters. What, in your opinion, are the 1 or 2 top priorities that we ought to be asking of our legislature in our annual reports from the CAC?
Ooh, that's a challenging one. I guess the only thing I can think, it's just making it so we can use this program efficiently. That the idea was that this was a regional program, it would be more efficient. And so sometimes it is actually pretty challenging to meet all the QIP funding policy pieces. So just if there's any way to have it be more streamlined and efficient, which I think kind of goes back to what I was saying, where I wish that DEC would say, hey, strategic wood addition is great. Go ahead. We're going to say, yes, do it. We can't guarantee right now what your credits will be. If you're willing to take that risk, fine. If you're not, then wait, and we're going to develop a methodology. So just making it so we can keep, like, we have more funds right now than we have applications for. So just making it easier to have people apply, and, opening up the project types to more types of projects would be helpful.
this may be an unfair question, but is that a question for the legislature or a question for DEC?
Probably more for DEC. Yeah.
Yeah. Okay.
Yeah.
I do have one further musing, and that is, again, I have not followed the nuts and bolts of what the TAC funds, the Basin Program TAC funds in the area of implementation. I'm just curious whether anybody else knows kind of the intersection of how the Basin Program is using its funding in the area of implementation and how that's kind of, is that duplicating any efforts or is that complementing efforts in the CWIS? And that's not a question of you, Hillary, necessarily. Just does anybody really kind of know?
That's a really great question. And I defer to others to answer it, but I do have like just something to add to that because I do know that there were conversations about like competition for phosphorus crediting between there was a dam removal project and I was not the lead, so I really can't speak to any details, but that was needed to be partially funded by Lake Champlain and then was asked to be also funded by the QUISP. And because of the shared of the phosphorus crediting, that project couldn't move forward with a shared funding source.
So that has been updated.
Okay.
So the QUIP funding policy updates do include that if the QUISP is a partner on a project, the QUISP gets the phosphorus credits. And I think there are still a couple of partners who that doesn't apply to, but Lake Champlain, the LCBP, it does apply to. So if there's a partnership there, the QUISP would receive the credits. Although any project you do with Lake Champlain Basin Program, those credits will eventually go to DEC. So if it's solely funded by Lake Champlain Basin Program, the credits would still eventually go to DEC. They will report those kilograms. Or sometimes they can be shared, like a dam removal, like you mentioned, could have multiple funding sources.
And that would be the case. So that's good to hear that happens even if it didn't meet the cost efficiencies that DEC would have liked to have seen.
Yeah, so we had a project like that where it was a really expensive project, and the main goal of the project was fish passage, but we were short on funds, and so we basically applied to our QUISP. We had a kilogram of reduction, so we applied for a kilogram worth of funding, basically, and that helped get the project completed. The QUISP, at that point, we didn't realize we were supposed to share with LCBP, so our kilogram got cut in half, but I'm wondering now if we're going to get our half kilogram back. But yeah, that does happen. So sometimes there'll be a bigger project and the QUISP just basically says, here's my formula, I can put in this much.
So Karen's got her hand up.
Oh, sorry, Karen. I just saw you now.
So then to add to Hillary's explanation, other ways that we coordinate is that there are sometimes projects that we know Lake Champlain Basin Program is funding, like the Lake Watershed Action Plans. And so then the Clean Water Funds won't fund those. I think there's something in the Clean Water Fund policy that basically says if there's another source of money out there, then use that and don't use our money. So this is where Hillary was talking about, is if it's really expensive and we need to collaborate and use a lot of different sources. So I don't know if that answers your question, Breck, but we do talk, I think, between the two funding sources to just make sure that we're not both funding the same thing, because that becomes difficult because are not always funding the same thing, because especially when we're trying to figure something out, like how something's going to work, likewise action plans were new, you sort of want one group funding it initially maybe and trying to work out all the wrinkles. At this point, it more funded, but are more I think Clean Water Funds does fund it now, but in the beginning it was like that. So
but from the point of view of being able to report back to EPA, DEC can claim those credits and towards the overall reduction targets.
With what Hillary was saying, yes. With what I was more action plan, so that doesn't have the phosphorus credit associated with it. But I do know that we've been working with Lake Champlain Basin Program to figure out how to bring credits over to our end or our side. And you'd have to talk to CWIP to really get that answer.
I mean, I'm not asking to inflate our credits, but I think that we want to be able to demonstrate that we are making progress toward legitimate progress towards the targets that have been set.
Yeah.
And NRCS reports phosphorus too, so they work together with the state as well.
Yeah. I bet Sarah
and I
has
saw Paul.
Better
oh, sorry.
Yeah, Sarah has a better answer, I'm sure.
I was just going to try to help clarify. Yeah, so can you hear me okay?
Yep.
Yes.
Yeah, so in our Clean Water Initiative performance report where we report on our progress towards the phosphorus TMDL, we absolutely have several federal and state partners to help report on our phosphorus reductions and meeting the TMDL, like including the Lake Champlain Basin Program or NRCS, Agency of Ag, BHCB, other partners. And then what Hillary's talking about for the QUIS is like a little bit more sort of in the mechanics of how the clean water service provider reports on progress on their own grant progress towards meeting their own phosphorus targets. And so there's just a little bit, I don't know that I'm totally getting there and explaining this, but that's more sort of the phosphorus crediting is more related to when there's a project that has multiple funding that the QUIS can count the phosphorus reduction of that entire project versus just the proportional part that's that they funded. So that's how they report on their own performance. But that has all, regardless of the split of co-funding, that has, when there's those reporting partners, that's always been accounted for, to answer Breck's question in our phosphorus reduction reporting towards our TMDL.
Thanks, Sarah.
Well said. And Paul, you asked how we discovered the projects we adopted. So there's a watershed project database that lists all of the projects that have been implemented with state dollars. And so projects that are in that database that have a project ID number you're able to adopt those projects. And then Pulteney Meadow, we did a lot of project implementation prior to the QUISPs. So we have a lot of sort of firsthand knowledge of the different projects that are in our area. So we did a search of the database to gather the projects that were available, and then had ones that we knew needed some maintenance. And so we targeted those first so that we could help bring those back and make sure that they were working properly. And then Bob? Oh. Bob, did you have a question, another question?
I really don't. Sorry, I had some trouble getting off mute. I really don't have a question. No, it was an excellent presentation, and it just shows how difficult it is for every sector to meet their needs, and But
yeah,
I wouldn't mind just throwing this out there.
The threat was
I
there
mean,
that if we didn't do something like this, I think that EPA was looking at having to really improve the wastewater sector.
Yeah, that's exactly what's going to happen, because we're the only ones under 1974 Clean Water Act, because they couldn't get the Midwest farmers to possibly senators to possibly go on to it without exempting farm zone as a discharge. Just to give you a minor thought, I don't have the data from way back but before phosphorus reduction started around 2002, 2003, we're taking about 40,000 pounds in and from 40,000 pounds out. Last year in 2025, I took in 85,223 pounds for my two facilities for about all of Colchester and all of South Burlington, and I put out 428 pounds of phosphorus.
That's amazing. Yeah.
Yeah. So, I mean, that is what, obviously it came down from 65% in the 80s of phosphorus input to wastewater. to 3%. But yes, and in the end, if you aren't all successful, they will crank me to minus. I mean, what else are you going to do? They're going to put me to -0, and I'll pay all sorts of fines and fees for things that are only negative. I mean, because I can go to zero. I don't, Anyway, that's my rant. I'm just sorry. Right? I took an 85,000, put out 428, and um, that's kind of my rant. When I started there, they were taking in 55,000. They put out 2,200. In 2015, I cranked it down, and I'm spending about. And just give a reference on that. I'm spending about 300,000 on and about 2 million in infrastructure or less a while ago, and about 300,000 a year for those. That's the O&M costs and whatnot. So and chemical costs. So that's my rant. Sorry.
Thanks,
Thank you.
Bob.
I just wanted to educate some on how this goes.
And I just saw too that Chris Rotler, who's here, is the grant manager for this program. And he is reminding us too that it's not just the Lake Champlain Phosphorus TMDL, but also Lake Memphremagog. So I don't think I said that in my presentation.
I have one more quick question, Hillary, and maybe you don't know the answer, but do you know if there are some dam removal projects that the were originally we thought were supportive through the QUISP for funding based on the phosphorus crediting, and then we applied and it wasn't actually what we thought it would be. It was substantially less. Do you think that has changed now, that we should then reapply in certain, has it been updated to recognize the co-benefits, and has that changed fully into effect, or when will we know when the right time is?
I would say it hasn't changed significantly in terms of what's valued, that phosphorus is still that's the thing that this grant is trying to reduce. But the numbers have changed significantly. So it might be that your 1 or 2 kilograms from your dam removal are worth more now than they were worth. And so it might be worth having that conversation with your QUISP.
Okay. Thank you.
Yeah.
I had one more question, sorry.
Okay.
Project development, the examples you mentioned were stormwater and stream geomorphic assessments. I was just wondering, could project development also be scoping for dam removal projects? Could it be more expansive than just those two, or were there just 2 examples?
So I would look in the QUIP funding policy, and it has the assessment types for each project type. And then I would talk to your QUISP, because I think you could probably make it work.
Okay.
It might depend, maybe you diversify it out a little bit and look at strategic wood addition and also dam removal, culverts, strategic wood addition, dam removals or something. The stream project identification is a little challenging because the geomorphic assessment is listed as the project identification tool, and those are expensive and a little challenging right now because phase 1 is hard to do because it uses really old software. It's still using ArcView 3.2 or something, so it's challenging. It's not supported very well anymore. But I think the Rivers program is trying to improve that. So it's a little complicated because the stream project identification right now is this geomorphic assessment is listed as the type of assessment you need to do, and that's challenging. So I think working with your QUISP to see what your QUISP and your basin planner really to see what you could maybe work out, maybe your Rivers program person too. Probably the 3 of them.
This leads me to my last question, I promise, because I recognize time. And so the other thing I thought that was great about your QUISP is or your Basin Water Quality Council, is that you have a little bit of flexible funds. And I was curious if other QUISPs had adopted that, or if there's a meeting of the QUISPs, or whether some of these lessons are being translated to other QUISPs.
Yeah, I would say so. We were hoping that model would be adopted by the QUISPs. We do contract directly with the contractors. So in a way, somebody would bring us a project idea, and when we would write a contract for that, say, a consultant to come look at it or an engineer to come look at it, So what I don't, and that was a little controversial in the beginning. Like we were the first ones to do that, and nobody quite knew what like projects to put, what project type to put that under. It ended up in project development. So what I think would be helpful is like if my goal originally had been that a watershed group could apply and get their own pot of these project development funds that were a little bit not as specific as like a stormwater master plan, just allowed you to figure out like what funding source is this going to go to. And they're not very much, we started at $1,500 per project and like one field visit and some desktop work by a consultant. And it's been increased to $2,000 mostly because the consultants gave us these like amazing deliverables. They did a lot more work than we expected them to, and like just couldn't not do that. So I think again, seeing if your CLISP would be willing to sponsor something like that for your group or for multiple groups, or to do it themselves and then allow you to just tap into it. Like, could you hire somebody to visit these sites?
I love that. Thank you so much. Any other questions for Hillary before we move on?
Thanks, everybody.
That's great.
Thank you so much. And please share your slides deck. Oh, Denise, you look like a fashionista.
Just cold. My son is napping inside, so and it's so nice out. I'm like, I gotta sit outside.
I love it. You must have sun where you are because there's no sun here.
Oh, that's beautiful out.
Oh, really?
Yeah.
Okay, so last on our agenda, and recognizing that we're a little bit over, we have MOU updates, and then we have the retreat input. And I really would love to focus on and I think the other thing that is shown in a diagram versus a note is just the term expiration dates, and waiting for, I guess, to hear more on updates from DEC regarding our term updates. So that's something we need to keep as a reoccurring, because excited to have Jane join us and Evan as well. So that's another thing. But maybe we can just do like 2 minutes on the MOU and then go to the retreat if that works for folks. And I know the other thing was the summary meeting minutes, which I think we could table that till the next meeting if people were comfortable with that. But if there are any edits that you have to the meeting minutes, to please share those with myself and Mary, and we can revise that. Does that sound like a good plan? Okay, so MOU updates real quick is that we had the Lake Champlain Basin Program Steering Committee was April 14th and 15th, where we discussed the MOU more, and the result of that was that a survey would go out to voting steering committee members to basically give their input on 3 parts of the MOU. And I'm just pulling up that so that I can. And those parts is the term of the MOU, the composition of the HOAC, which is the Host Organization Advisory Committee, and the role the steering committee should have over staffing on the HOAC. So there's the survey that's out that voting committee members have to fill out. So that is happening right now. What's also happening is that, the small SAC received our comments related to the MOU and other people's, and they were going to, prepare an MOU, I think maybe based on this survey and the comments that they would present at the June strategic steering committee meeting. So that's where we are, and then I can also just share and I'll let other people add that the small group of us working on this from the Vermont CAC, also Breck helped draft a 3 points, like just 3 key points of the MOU that I need to circulate with this larger group. I'm realizing that I have not, so I will share those 3 points. It was shared with the steering committee meeting, but just as a way of like the 3 points that we are just have more questions about, and those are reflective or in line with those survey questions as well. And then Mark is here, but Mark drafted an op-ed that I think is soon to be released as well. And I'll leave it at that. And if anyone wants to weigh in, please do so.
That was a good summary, Karina.
Okay, great. Awesome. Then let's go to the retreat agenda. Because I really could use help with, and it is, we have a month and a couple weeks before the retreat, or a week. So I wanted to reach out to speakers or give Mary time to reach out to speakers. So I just wanted to talk about what we wanted to prioritize for our June 22nd retreat. And I think some things that, what last year we had the legislative update from Senator Watson and Representative Sheldon, Chair Sheldon. We could certainly do that again if folks thought that would be valuable. We could, and then if we do that, do we want another guest speaker? Who would that guest speaker be? Or do we just want to have plenty of time of our own to reflect on the legislative session and timing and future speakers, and also hear some updates from Lake Champlain Basin program?
I guess I think if you think about the purpose of the retreat, I'll just jump in if that's okay,
Yeah,
Karina.
please do.
Did I raise
Please.
my hand? I think the purpose of the retreat is really a wrap up, right? Like we've been doing kind of the what worked well, what could have been improved kind of from our processes, and how we're showing up and what we're doing. And then I think it's also, and I think it is an opportunity to hear from the legislative representatives if they're available. I found great value in having Chair Watson and Sheldon there last year. And again, I know it's timing, and if they can make it, I think that's really nice. And then I think the third piece of it to me is, planning for next year, right? Like, what does the CAC want to prioritize both in our educational opportunities? Like, what do we want to learn about? And then what actions do we want to take and make sure are incorporated into our action plan for the future? And so I think it's an opportunity for us to plan for the next year, starting in June. That would be, well, especially if we're, again, because, our action plan then really starts in earnest kind of after that meeting, where we start thinking about what we want included in that action plan and how we might want to adjust or change it. I heard, sorry, and I don't know if we've talked about this already, that the chloride bill was vetoed by the governor.
It was.
Okay.
Yes.
Yeah, I
Yes.
saw Jared and he
Yes.
looked a little distraught the other day. I was like, oh, yeah, that's too bad.
I thought we totally got that. When we met with him, it was real obvious as we talked to him while we met with him that he was going to veto it because of tort reform. Thank you.
Yeah, right, because of the legal stuff that he was worried about. Yeah. Anyways, but that's the kind of stuff I like to hear about at the retreat. Like, I like getting kind of the inside baseball stuff or what happened, and then also any other policy. And for education at the retreat, I don't have anything really pressing for me right now, I don't think, but maybe other people do.
Lori.
I agree with everything Denise raised, and I think with the if we are able to get the legislative chairs back to do presentations, I think that we want that very directed to our priorities. And, specifically, for example, with the chloride bill, is there something, that the CAC should be doing or working on to head off another veto? I mean, the governor noted that and that there wasn't enough protection to for um, municipalities and contractors, from his perspective, and the Vermont League of Cities and Towns felt the same way. So is there something that we can be running interference on? The counter to that is that doesn't stack up with a program modeled after the New Hampshire program, which was been going since what, 2013 or something. And so, what can we do to have a better performance success on with regard to the our action plan priorities? Another thing I'd suggest we do is, we've had a lot of presentations this year, and maybe we want to spend some time thinking about what came out of those presentations and some questions that can inform our priorities. For instance, last month we had the tile drain presentation, and I know some of us have worked on tile drain legislation in the past, and just getting, and for 10 years trying to get a mapping and an inventory of existing tile drains. And so for me, I see that as, that was one of the questions I raised last month, and found out that no, we really still don't know where those tile drains are, how many we have, how old they are. So requiring tile drain mapping is, a priority for me, coming out of these presentations that we've had during the year. So maybe since we only have a month between now and the next, in our retreat, maybe we can all be pulling, reviewing the notes from our meeting and the presentations, and coming up with some ideas to help build the action plan agenda, because that's, part of what we dive into and try and um, uh, structure a framework for a bit at the retreat.
I love that.
Yeah, I love that too. That's a great idea. Even if we could have like Claude or something kind of or
Yeah.
someone take each of the presentations and just summarize, that we could like have like a handout that day to be like, on this day we heard from this person, and these people, and this was the summary of that conversation. And then we could all be thinking about questions or actions that we want to take. I love that idea, Lori. That's great.
I guess my thought is that that the retreat is where we can also have these honest conversations about what what's working, what are we reaching people? Are they using the information we give them to influence a decision they make in the legislature or in the agencies? And are there examples of where that's worked, and can we continue to improve our ability to actually influence outcomes? And I know it's not an easy thing to do, but I think it is pretty important to think that through.
I like that. Okay, so I'm just looking at the sort of a draft agenda, and we can circulate this. Mary sent it to me this afternoon. And I'm wondering, probably, I think it would be the morning. I guess it's really availability of the chairs of when they could come, but I think how we did that last time was in the morning. So I think that was like a a 9:30 or
They
10
came pretty
o'clock.
quick right after we started.
Yeah. So we can defer to them on that, but I think we can go ahead, Mary, and ask them to attend. If you could do that, that would be great. And then we can work on our agenda around that with, I think, a lot of the time that we just set for reflection and summarizing, synthesizing what we've learned with the presentations. Lori.
And we also, we've been spending a lot of time on the MOU. We don't quite know what, where that will be, but we had our subcommittee had asked Mark, and Mark, I think you're at this meeting now, to participate in the CAC retreat as well, because and so I would say the MOU, probably we want that as an agenda item too, just because I think that's such an ongoing issue, and the CAC, the Vermont CAC in particular, has been playing just such an important role in that discussion and deliberations. And so we want everybody to know that Mark is one of the invitees to this meeting. And that's, part of the reason, as well as his historical involvement with the CAC and kind of knowledge of these issues too.
Yeah, thanks, Lori. I think particularly depending on what happens between the survey, the June meeting, and the setup for a potential vote on what that MOU might look like in September, I think it'd be good to have that conversation. I don't want to take too much time, but yeah. And I just, one other thing that I've been thinking about is how well the CAC is doing outreach to engage with the citizens of the state so that we're addressing the issues that are important to them. I know it wavers and we've always sort of struggled with it, but I think it's a good thing for us to remind ourselves of
our charge.
How can we do better with our charge? Yeah. And budgeting through the agency in order to create better outreach. We're more technologically connected, but we're also less informed. I think are less meaningfully informed. I don't know, that my judgment. So how we might be better about that, I think what has been a consistent thing that I think for all of my time on the CAC, we've been trying to figure out how to do better. And I think it's always good to bring back up at the retreat.
Yeah, I think that's a great point, Mark.
I love that.
Yeah. I don't think we send out enough press releases about the work that we're doing or getting people, although we had a lot of people tonight, public at our meeting tonight.
Yeah, it's sort of an issue oriented. Sometimes there's lots and lots of engagement, others, and we didn't participate in the Act 181, Act 250 stuff as strongly, but clearly there was a misread on what people thought about it as Tier 3 was getting rolled out, et cetera, and there was a full retreat on it from the legislature. So
I think that would be an important part of the legislative update. Chair Sheldon and Watson have both been incredibly involved in that. So, but yeah, huge misread on what Vermonters wanted.
Mm-hmm. I love that idea.
The most important thing.
What?
A clean lake is the most important thing. We can all agree. And speak for nearly everyone, right?
I had a thought and it went out the window, but I think those are all really good things. And there's probably not enough time in our day to cover them all, so we will have to use our time wisely.
I've waited till
I mean, I can help draft the agenda with you if you want me to. If you
That
want
would be great.
or we could do it asynchronously.
That sounds great. And oh, I don't remember. I was just
You said and then
oh, I know. Mark, it came back to me. Can we share or could you share the op-ed with the group or with me to share with the group when it goes out? That was my other thought.
I thought you emailed it. Am I misread? Did I
I
get
mean,
the
I
wrong?
have the op-ed. I just didn't know if you have released the op-ed to release the op-ed. You know what I'm saying?
Mark, you're muted.
Still muted.
I think let me wait till it's released, and then I think wait till it's released.
Yeah, I think and Mark has an opportunity to have some conversation with where it's released too
Right.
before because otherwise you don't want it to be inadvertently circulated.
Right.
Yeah, so I will defer to you, Mark, to just let us know or let me know and I can circulate it at when you're comfortable.
Okay.
Awesome. Dinner is served at my house and
Do you have a butler?
I wish I did. So, if I could have I don't know if we made a motion to start this meeting, but if someone wanted to make a motion to
I'll make a motion.
So moved or second.
Thank you. Great meeting, folks.
Thanks for organizing it. Thanks, Mary, for all
Good
your
to see you all.
hard work behind the scenes.
Thanks, everyone.
So.
Bye.